Hello again, students. I'm recording this again, being quietly judged by the workers in my game studio. They're very judgy, but I will persevere. This video, I'm going to show you experiment mode, and I'm going to play through 10, 15 generations of one game, and then we'll have a game's worth of data, and I can show you how to start visualizing it in R. So we're just going to play through one long game. We'll try to squeeze it in before they're coming to shoot a commercial in this lab pretty soon, but I think I can get this done. So this is the start screen for Darwin's Demons. I've cleared out my data files and all that kind of stuff. And I want to just show you experiment mode because the bulk of your data are going to be collected in experiment mode for a variety of good reasons, which I will describe now. To get to experiment mode, you can use your mouse or the arrow keys or whatever you want to do. You click on mode, and it'll say experiment under there. And you can see it grays out the difficulty selector now because experiment mode, you have direct access to the parameters that actually we use to tune the difficulty. So remember, you want to go to options and make sure record data is checked. I described that in the last video. Now we're in experiment mode. Then you click start. This takes you to the experiment mode screen. So there's a variety of game parameters that you can play around with to run experiments in Darwin's Demons. And some of them are really useful, and we use them a lot in Biology 315, and others are more for playing around with gameplay and not particularly useful. Okay, so I said okay again. I'm trying not to do that. I'll just walk you through the various sections here. First, we have game parameters up here at the top. Mutation rate. So mutation is a function of this game. It's how we introduce variation into the genomes of the creatures upon which selection can act. Mutation rate can vary from 0, no mutations, to 1. Mutation is the probability that a given gene will mutate per individual per generation. There's roughly 50 genes, I think, in the genome of Darwin's Demons. So probability that a given gene will mutate per individual per generation. And so you can vary that from 0 to 1. This probability in real cells is very low, 1 in 1 billion. Our mutation rates, to make it a fun game to play, are ridiculously high. It's just a piece of the artistic license that you indulge in when you make an evolutionary video game. So we're going to leave the mutation rate to be 0.1, which is still insanely high. Standard deviation. This is essentially the effect size of a new mutation. Essentially just think of it as higher is large effect mutations, lower is low effect mutation. It is a unitless parameter, so I can't really describe it any better than that. If you crank it up to 10, you will get some crazy high mutational effects. If you crank it down to the basal value of, whatever this is, like 1 or something like that, it's reasonable. So let's just set it at 1. Tournament size is the size of the tournament that we use when reproduction occurs between generations. And so tournament selection is a technique that computer scientists use in evolutionary computation to select individuals to reproduce. And so how this works in Darwin's Demons is after every generation, we conduct a tournament to see who will win the tournament and become parents. So every time we need to identify a parent, we randomly draw tournament size number of individuals. In this case, it's 20. We randomly draw 20 individuals from the population. We order them in terms of their fitness, and then we pick the best one. And we say, we set them at an individual. That individual is going to be a parent. Well, they need someone with whom they can reproduce. In our game, these are hermaphrodites. There's no males or females. We do another tournament, size 20. Grab 20 individuals. This is the best one, the most fittest one. We'll get to fitness functions in a second. Put it over here. Now we have these 2 individuals. They have a baby. Everybody goes back into the pool. And we keep conducting tournaments like that until we have created the number of individuals in the next generation. That's, I don't know, it varies. You can control that actually. Recombination is a toggle. You can turn it on and off. If it's unchecked, the offspring get one chromosome from each parent. All the individuals are diploid. If checked, all of the loci recombine freely, and they behave like they're on separate chromosomes. We have not played around with recombination much in Darwin's Demons, so just leave it checked and I don't know, you can play around with it as part of your experiments if you want. That would be a fun experiment. What does recombination do? I don't even know. Waves per generation is essentially a way of controlling population size. Each wave in a particular generation adds 13 more aliens, proteins. So if we add 10 waves per generation, our population size is much bigger than if we have 0 additional waves per population. So that's essentially 0 is just the first wave and that's it. I'm going to just put that at 4 because I kind of want to play through this quickly. Anomaly rate. So, remember Space Invaders? Did you ask your dad about Space Invaders? Did you? I told you to. Did he kind of geek out? Anyway, probably not. Anomaly rate is like the boss aliens in Space Invaders. And anomaly rate is the probability that a genetic anomaly or a really crazy, highly evolved boss alien will migrate into the population. When we're doing experiments for Bio 315, unless you're particularly curious about anomaly rate, I would say turn this to 0. Let's get rid of genetic anomalies because they're like migrants and they mess up our analyses of selection. So, all right. Now, initial creature genomes, we're going to not really play around with this in this video, but you can start with creature genomes that are sort of pre-evolved to have certain behaviors. This is about the starting standing genetic variation. Let's just leave it at defaults for now. Okay? But you can, if you want, to start with creatures that are already pre-evolved to charge the player. You would just click this and that's what you would start with. Or creatures that are pre-evolved to avoid the bullets and so forth. Fitness components. You can evaluate fitness using 3 different variables in Darwin's Demons. Aggression is how far they get down the screen. And if they go off the screen, you lose a life and they get a bonus to aggression fitness. Accuracy is how good they are at shooting you. The closer they come to shooting you, the more fitness they get. The more shots they shoot, the more fitness they get. And then lifetime is how long they live. I highly, highly suggest you uncheck lifetime for your experiments. I'll make a whole separate video about this, but lifetime had some unexpected effects when we encoded this into the game during development that created some really funny stories. So let's just leave aggression and accuracy checked for right now. Don't worry about max bullet speed and starting bullet speed. You can adjust those if you want, but they're more like gameplay difficulty parameters. The last thing with experiment mode, that's super, super important. Darwin's Demons is actually quite difficult. It's a hard game, especially for new players. And you can't really collect a lot of meaningful data if you start with 5 lives and you're dead by generation 4. So, I suggest, whenever you start a replicate, you start with a fixed number and a high value for this field, starting lives. I suggest 100 is okay, but if you really suck at the game, 1,000 is fine. Just make sure it's the same number, because sometimes it's fun to track the decay value, your mortality rate, how many lives you lose over time. Okay? So I'm going to go ahead and I'm going to give myself 100 lives. Now, other things that you can adjust if you want are starting biomatter, biomatter per kill, biomatter kill scale. This allows you to upgrade faster if you choose to incorporate upgrading into your experiments. I kind of don't really do that all that much, so I'm going to leave these blank. Okay? So, I'm going to start with 100 lives. I've got these parameters set. I've turned the anomaly rate off so that we can study evolution by natural selection instead of the effects of weird migrants. And then I hit confirm, and then it takes you to the standard starting point for the game, where you pick your ship and all of that kind of stuff. So I am going to just play, and then we'll accelerate the gameplay. I am going to choose the Manta Ray. That's a fun one. It's pretty powerful. I'm going to choose something that I don't have to remember to use. So I'm going to choose these orbital pods that shoot straight. I don't like the ice aliens, so I'm going to choose an ice sword that automatically zaps the ice aliens. And I'm going to choose increased fire rate. And then I'll hit launch. And here's what I'm going to do. I'm going to play for like 10, 15 generations. And we'll accelerate it and maybe set it to music. I don't know. Like Benny Hill clown music. Anyway, here we go. Okay, I think 18 generations is enough. So I'm going to have to pause this again because we had people come in and shoot a commercial, and now we got somebody coming in who is late, who thinks he's going to be in the commercial, and I have to tell him he's not going to be. Okay, I'm back recording again. So I played. I didn't lose all my lives. I played for 18 generations. That's typically what you do with this game. You'll collect data for 15 to 20 generations, and then you'll stop. And I just want to sort of orient you to kind of what happened, and uh, and then in the next few videos, I'll show you how to visualize the data from a single run of the game using R in RStudio. So you might have seen me upgrading in between generations. You can do that or not, uh, for according to what you want to do with your experiment. But remember, you have to do it the same way every replicate. So you should make it simple. Don't make complicated choices for your upgrades that vary across replicates. We want to hold as many variables as we can constant, um, so that we can analyze the variable of interest. So, but in this case, we're just doing one run. If you want, you can look at, so this is, after 18 generations, this is survival of the fittest. This is the 5 fittest aliens. And you can see most of what they're optimizing is what we call aggression fitness. So they're trying really hard to get down on the bottom of the screen. Lifespan we're kind of ignoring for now, but this is how long they lived. And then accuracy fitness, you can see this individual was the most fit of these 5 because it was able to shoot me a little bit. These graphs here, type shows you the evolution of the different alien types over time. Generation is on the x-axis, the different frequencies of type. So gray is like just the generic alien. This is like the alien that shoots in sine waves. This is the alien that shoots with fire. This is the alien that shoots directly at you with acid. This is the psychic alien. You see, we had very, very few of the blue aliens because that's why I had that blue sword that automatically shot blue aliens as soon as they showed up on the screen. Right? Different shield types. That's a pretty dramatic effect, right? So what you should know is the kind of weapon that my ship has, which was a laser weapon, is particularly weak against this kind of green shield that the aliens evolved. And so, not particularly surprising that the frequency of green shields proliferated throughout this particular replicate. The rest of these, dominance, fitness, enemy fire rate, avoidance, charging, I actually would rather show you guys how to graph those in R. We use these sometimes when we talk to high school kids and they don't have a ton of time, but we want to do a deeper dive into the data, and so we'll ignore those kinds of things. What students have done in the past is they will try to use these graphs for each replicate, but like when you do a new replicate, what are you going to do? Take a screenshot? Like, no, we want to do this more quantitatively so you can compare across replicates what happens. So let's just say we're done. We decided we would go 18 generations. If you hit main menu, you're going to go back to the main menu, and the game will have recorded the data. Okay, so let's have a look at what that looks like, and then I'll stop this video and we'll record more in terms of how I analyze it. I go to options. I'm like, I just recorded a bunch of data. I would like to see it. Again, options, and because I'm screen recording, it's laggy. I've opened my data path, and there it is. It's almost a meg, and the last thing I want to do is I just want to show you why doing this in Excel is a huge mistake. So if I double click on this, my machine will open it in Excel, and it's going to take a while because it's a big data file. It seems to think Excel is already open. See, I get the spinning beach ball of death. Okay, so remember we had 60 variables, and if I go all the way to the bottom of this data file, I had 1,483 aliens. So you've got yourself a spreadsheet here that's 1,483 rows by 60 columns. And we're going to have to do multiple replicates to conduct any kind of an experiment. And so you very quickly exceed the capabilities of Excel to be useful to you in any way. Okay? So I'm going to show you how to deal with this stuff in R. And in the next few ones, I'll tell you how to get R and ingest this file so we can have a look at it and see what happened to our aliens when I shot at them with lasers for 18 generations. All right, I'm signing off. Landon, do you want to say anything? Nope, Landon, no, the heck with you.